So I'm Lainy. I'm from the University of Mississippi. Nobody knows where that is. It's Oxford, Mississippi. It's about an hour from Memphis and about 50 years below the Mason-Dixon line. So I would like to understand how patterns of selective forces lead to adaptations in brain hormones and behavior. I've been looking at this for years in species as diverse as frogs, lizards, lowly humans, etc. So first, when we talk about this, we have to understand behavior. So using high-speed video, we have been looking at a lot of the different manakin species, and one of the things we discovered is that the poor, dumpy black manakin that had absolutely no display actually does a perfect back flip with great precision in about 360 milliseconds. So in real time, this looks like a really simple hop. You can't see it with the naked eye. We also found that the moonwalk of Pipper mentalis and the moonwalk of Pipper cornuta is actually performed differently. Mentalis does these tiny little hops, whereas Pipper cornuta does this sliding step. The differential mechanics alone raise so many fascinating questions. But back to the brains we're stepping to look at. So for 13 species in the 8 genera shown, we developed a complexity score for the illustrated displays. The complexity scores are given here. We used the discrete characters that were published by Prum, adding all sorts of recent data from those of you who had done other observations, and then our own observations with the high-speed videos to establish the scoring system. And our previous work with the golden-collared manakin showing that there were differences in these motor regions between males and females, the volume of these brain motor regions between males and females, certainly influenced our hypotheses about the entire clade, which have now been supported. We've shown that manakins that have the most complex splays have the most complicated, have the largest brain relative to body size. We have since replicated this with brain volume and have preliminary data that's also showing that the archipelagian volume and the cerebellum, features of the cerebellum are related to the display complexity as well. And as we began, I'm also interested in looking at the neuroendocrine system. Thus, we have been working to determine the relationship between androgen receptor, estrogen receptors, and aromatase in the brain. And as we began designing primers for quantitative PCR, we found that there were nucleotide substitutions in our genes of interest between the manakin species that were in our primer areas. We took advantage of the published genome in this work, and because we were looking at 13 species rather than 2 or 4, it was essential that our primers be designed for a region of the gene that is conserved across all the species. Otherwise, we wouldn't know whether it was due to our primer inefficiency or actual differences. So now we have sequenced for 13 species, 3 of the 4 genes that we're interested in. And lastly, we've also obtained a whole transcriptome for the cerebellum, and we'll be working with Chris to find out if we can see expression profiles that are interesting, and also just to add to our ability to annotate the genome with these transcriptome data.